Thank you for the introduction. Yeah, I'm going to talk to you about how we're really using our tech, our DNA tech at Twist, to build these antibody libraries, precision antibody libraries, and how we're really leveraging that DNA tech. So there are a couple of products that really work well for precision antibodies libraries. One is our variants library, and I'll just touch on that as a product and show you some of the advantages of going with the variant libraries if you're looking to design such libraries yourself. And I'll talk about a case study of how we've used this particular product to build precision antibody libraries. But I also want to focus on a new product that's come out of Twist called Multiplex gene fragments, which is essentially a pool of gene fragments where we can build, for example, exact V-gene sequences as a pool. And I've got a case study here looking at de novo rationally designed VHH genes and how we've helped to build such libraries using the multiplex gene fragments. So to start, let's talk about the Twist variant libraries. So it really does start with how we at Twist are building our DNA in the first place. We're using phosphoramidite-based chemistry, but we've miniaturized that chemistry down onto a silicon platform. And using microfluidics, what we're able to do on a similar sort of footprint as a 96-well plate is build 1 million oligos with base-by-base precision up to 300 nucleotides in length. And having miniaturized this process, we use very little raw materials, and that makes it extremely cost-effective, and we're passing that cost on. But the advantage of using the platform we have is the quality of the oligos that we end up with. We get extremely high uniformity, as shown by that type distribution on the left. We get 100% oligo representation, so no dropouts. And we have the lowest error rate in the industry of 1 in every 3,000 bases. So it's this high throughput, high quality oligos that really is our engine at Twist and what we use to build custom products further down. So what can we do? We can assemble the oligos to make gene fragments. We can clone those into a vector, your vector or our vector, to make sequence-perfect clonal genes. What I'm going to talk to you about is how we're using these high-quality oligos to build high-quality libraries. Now, there are many different ways that people are building libraries. You can use degenerate oligos, NNK, NNS, a trimer-based approach, or you can use what we do at Twist, essentially. And what's different at Twist is we will take your library design and we will work out exactly what oligos we need to synthesize to precisely build that design. And that means that once we've constructed our library, a Twist-constructed library is very, very similar to your design. In fact, plus or minus 1%. With some of the other approaches, what ends up happening is that the constructed library is a distance away from your design. You end up having variants that you haven't actually designed within that library and biasing what you see within those libraries. So what it means is that you end up having to screen your library more to find your variants of interest. Whereas with a Twist library, the variants of interest are within that library and in the proportions that you have designed. So our library product really splits out into 3 main products. Firstly, there's the site saturation variant library. In this case, you're changing one position at a time along your open reading frame to a number of other codons. The other 2 libraries are combinatorial variant libraries. So our sold library is where the diversity is scattered along your gene. So you have positions that are spread along that open reading frame that you wish to change to a number of other codons. So we can build our sold library up to 10 to the 8 in terms of diversity. There is a restriction on length there. We can't really go above 1.5 kb because of the way in which we build that library. But by far the most popular library for antibody engineering, antibody discovery, even antibody optimization, is really our standard CVL library. That's our combinatorial variant library. Here what we can do is we can build in diversity within stretches along the open reading frame, domains. These could be CDRs, for example. And then we can assemble these domains together to build really high diversity libraries up to 10 billion variants. So really the take home on why Twist libraries are different is our ability to print these oligos with base-by-base precision. It allows us to follow the rules of code on usage tables. You can choose to exclude certain restriction sites. We just in silico remove any oligos that encode such sites. You can choose to have sequence liabilities removed. That's, again, easy for us to do. Deamidation sites, glycosylation sites, we can just avoid those. You have a lot of control when you come to design your libraries. For example, if you wanted to change the ratios at certain positions and have different amino acid distributions, that's easy for us to do. We would synthesize the oligos in that way to replicate your design. And if you come to us with a list of oligos that you wish to see within certain CDRs, just actual CDR sequences, this is, again, something easy for us to do. We would just print those oligos and build that as an oligo pool and incorporate that into your library. And most importantly, I guess, is we do a good number of reads on NGS once we have built your library, and we'll provide you with a report where you can see what's observed from the NGS run and how it compares to your design. And you see how these can marry up really nicely and match up. So I'm just going to move on and talk to you about a case study where we've built these precision antibody library. It was a target-focused library, and we used some existing structural data to build it. This work was done with macromoltec. It was with a GPCR target, C5a receptor. And what macromoltec were able to do using existing structural data was identify discrete druggable epitopes and then predict antibody binding sequences to these epitopes. So in the end, they gave us a list of 10,000 putative binder sequences. We took this list and we built oligo pools of all the CDR1s, the 2s, the 3s, and assembled that into a library. And in fact, what we built was a single-chain FV library, so all 6 CDRs. Now, if you look at the theoretical diversity, and we looked at all combinations that could be made, we had an extremely large library of 1 times 10 to the 25. But what we ended up building was a representative sample of this of 1 times 10 to the 9. And the maximum size we tend to build our libraries to is 10 billion variants at Twist. So it's always good to consider when you're designing such libraries on what your theoretical diversity is going to be. So the closer you get it to the size that we can build and you can screen, it means that your variants of interest will be within that library. Now, from the NGSQC, you will get a lot of detail. One of the things that I've shown here is how the length distribution profile compares from our NGS runs to what was in the design. So in the design ones are in blue, and in red are what we observed from our NGS run, and you can see that they match up really nicely. So this library was then run through phage display. We carried out 5 rounds of panning with increasing stringency. We carried out some NGS sequencing after each round of panning to look for enrichment. And we also carried out some unsupervised clustering analysis. And using the NGS data and the clustering data, we were able to identify a good number of unique clones In fact, we found 45 clones with unique CDR3. And at the end of this, what we did is we selected 96 antibodies to reformat as full-length IgG. Reformatting at Twist is very straightforward. It just fits into our high-throughput antibody production facility. This is a standalone product. And here what we did is we took the V genes, the variable heavy and the variable light genes, we coded and optimized them, we synthesized them, and cloned them into expression vectors encoding the constant regions that we required. These were then transfected into HEC293 cells using lipofectamine, and after a few days incubation, the supernatants were harvested and protein A purified. We carried out some basic QC on the purified antibodies, a digital gel to give you an idea of size and purity, and an A280 read to give you an idea of concentration and yield. Now, this workflow is very automated. It's built with a set of Hamiltons. We don't necessarily have to use HEC293 cells. We can use ex vivo, but for this case study, we did use HEC cells. So, as I mentioned, it was a GPCR target. So the first point of characterization for this particular target was flow cytometry, where we identified hits that showed a threefold improvement in binding to C5a receptor cells. And from those hits, we carried on to do some titration on flow cytometry to confirm binding. But what was interesting is what we were able to do in this case is also use SPR to confirm binding. This was carried out on the CAR-T LSA system. Here, we worked with a company called G-Clips Biotech, who provided us with mice cells that were loaded with the target, loaded with C5a receptor. And we were able to use this on the CAR-T system to show binding and confirm binding of our hits. But for this particular target, the most important assay was a functional assay, where we wanted to see which of our hits were best at reducing beta-arrestin recruitment. And we found a set of leads that were almost similar to our competitor antibody, avdoralimab. So they were great, but not quite to the same level of potency as the competitor. So at this stage, what we did is we went on to take those leads into our Twist antibody optimization platform. So here, we look at those lead sequences, and we'll build a diverse library around those sequences, but considering that we don't want to move too far away from that lead sequence. So there's a lot of expertise, and we have a number of ways in which we design these libraries to enhance antibody optimization and affinity maturation. These libraries then went through phage display again with a number of rounds of panning and screening. We selected 96 antibodies again for each of those leads, and then reformatted those to IgG, ran through our binding assays, as well as this reduction in beta recruitment assay to look for potency. And in fact, with this antibody optimization workflow, we were able to improve the potency 16-fold. How does that compare to our competitor antibody? Well, we found some that were similar to the competitor, and some, like TB661003, that had an improved potency to the competitor as well. And just to say, in our panning rounds for affinity maturation, we were able to use cell-based panning, and we were also able to use those mice cells that were loaded with the target as well. And that certainly helped. So I'm just going to pause there and say, we worked with Macromoltech, who provided us with 10,000 antibody sequences. And then we went on to expand on that diversity and build a library of 10 to the 9. And when I've presented this in the past, I've often got a question to say, well, why didn't you just test the 10,000 putative binder sequences? Why then, when they've predicted that those are binders, did you go out and expand that diversity? There were several reasons why we expanded the diversity, but one of those reasons was to build 10,000 gene fragments to then pool into a library is actually not as cost-effective as building a library, a combinatorial library that incorporates those 10,000 sequences. So that case study predated our new product, Multiplex gene fragments. And perhaps if we had this product, we may have done things differently for that particular case study. So here, what we have are gene fragments that are not arrayed, but in a pool. And these gene fragments can be up to 500 nucleotides in length. So it can fit a V gene very nicely in that space. And it comes down to how we're synthesizing those oligos on that silicon chip. We can now push that length beyond 300 nucleotides, and we've pushed it to 500 nucleotides. So we're literally printing V genes as oligos and then amplifying them, making them double-stranded, carrying out some fragment QC, and at that point they're ready to ship as a library of exact sequences. So just to look a little bit at the spec, this is a really new product. It was only launched last month. We can take lengths of anything from 301 to 500 nucleotides. We do require constant flanks on the ends, 20 to 25 nucleotides on either end, to allow us to amplify and make these double-stranded. There's really no limit to the pool size, and it's a really quick turnaround. We can get these out in 8 to 12 business days from when we start synthesis. Prior to synthesis, there will be a little bit of back and forth just to ensure that the sequences are suitable for this product and just to ensure that we have the flanks and it matches, the sequences will work for production. We're kind of delivering about 200 nanograms, but this really depends on your pool size. So if you have a larger pool size, you'll receive at least 200 nanograms. We have delivered micrograms of this too. We have extremely high uniformity from the ones we've carried out so far, and the error rate is still holding quite high. So even though we're able to stretch this length to 500 nucleotides now, we're maintaining a really good error rate. So a slight drop to 1 in 2,000 nucleotides, but still the best in the industry, and we're the only ones really able to build oligos of this length. So how does this product benefit us? Well, as I said in the previous case study, we shuffled the CDRs. And this multiplex gene fragments means that we can actually keep trios of CDRs. We don't need to shuffle any of the CDRs. We can keep them exactly as they were intended in those three kind of three trios. And I'm just going to talk to you about how we worked with the Baker Lab at the University of Washington. They utilize our multiplex gene fragments to really test out their de novo design of VHH antibodies. So at the Baker Lab, they have an AI protein design tool that they were using to design VHH antibodies, and they designed 9,000 VHH genes to each of 4 targets. What they wanted was a pool of those exact 9,000 genes. They didn't want added diversity. It was really important for them to have just those sequences to test to see if their algorithms were actually predicting suitable binders. We were able to make these using the multiplex gene fragments products as separate pools of 9,000. These were then used by the Baker Lab in their yeast surface display platform to identify binders, and then a selection were expressed in E. coli and screened on SPR. And they were able to identify some top binders from there. So this information allowed them to really refine their algorithms, and from there they're now really able to predict antibody sequences that would bind to certain epitopes, and they're really able to design de novo antibodies. So I'm going to just summarize now that Twist can help in many different ways. It's not just genes and gene fragments. We can now offer these kind of multiplex gene fragments. We can offer oligopools or cloned oligopools up to 300 nucleotides in length. I've talked to you about the different library options. And we also have a Biopharma Solutions Discovery Services group, where you could essentially bring us your target of interest, your wish list, what you want to see in terms of binding affinity, what species cross-reactivity you'd like to see, what specificity you want to see, ensuring it binds to a certain target member and not to closely related family members, for example. And we can take the entire discovery service for you and deliver candidate-ready drugs for you at the end. So I'm going to pause there. Do come and talk to us. We've got a booth. If you have any projects that come in mind that you want to kind of bounce off some ideas, we're very much here and happy to help. So thank you.
Thanks for nice talk again. I might have missed, but so this is a synthetic way of generating a library. Has this been compared with natural way in the form of like polyclonal antibodies? You gave one example of monoclonal, I guess.
Yes, so often we have taken natural repertoires and built that as a library. So when you say synthetic, yes, these are built synthetically, but we can really follow the rules of what's in nature. So we've done that in the past. We've taken we can build immune libraries of what's coming out from in vivo discovery as well.
As efficient as natural ones, right?
Yes, so the design can be exactly what you see in nature, and we can replicate that.
Yeah, okay, thank you.